All right, in the last few sections of Chapter 12, we're going to talk about a couple of different viruses specific to humans, and they each have slightly different genome construction. We're first going to talk about the papillomaviruses. They're a DNA virus. And this is an interesting one because papillomaviruses can actually cause human cancers, and we'll see how that occurs. So let's start with a case study. We have Sean. He's a 19-year-old college sophomore. He goes to the campus health center and he tells the nurse practitioner that he has a raised spot on the shaft of his penis. It doesn't hurt, but it's not normal, so he's going to get it checked out. The practitioner tells him that it is probably human papillomavirus, HPV, which can cause genital warts. So how does it do this? So HPV strains can cause warts, and they're transmitted by direct contact. This could occur during sexual intercourse. So the practitioner is going to conduct a physical exam and find several spots also on the anus. So he has probably been having unprotected sex. The practitioner tells him that the warts probably will go away on their own if he maintains healthy sleep and eating habits. His immune system will rid him of these. But some HPV strains can persist for years without symptoms and still be able to be transmitted. All right. This is a medical course, but I'm going to give you fair warning. If you don't want to see the warty penis, it's not too bad, I promise, then don't watch the next few minutes. But you could listen to it and see the explanation of what's occurring. I swear it's not that bad, and this is a medical course. You're going to learn all about reproductive organs and their issues. So here we go. All right, so you can see on the shaft of the penis here that there are all of these raised spots that are darkened in color. These are growths that are occurring there. So what's actually happening? HPV has infected the epidermal cell layer, and it goes down to the basal cell layer, where it can lay dormant. HPV can actually integrate into the human genome and affect gene expression, turning the cells into cancerous cells. Remember, cancer is unrestricted cell growth. So these growths here are cancerous in nature because of the HPV in there, which is causing them to replicate. HPV is in the genome, so every time the cell replicates, HPV is replicated. This can lay dormant in these basal cells, but they can be activated and turn into keratinocytes, which are these thicker cells here. And in the process, the cells can release the HPV virions after this. So this is a common way that it gets transmitted. So he was likely in contact with strains that can cause cancer, the penis or the anus. In this case, of course, in females, the vagina as well is susceptible to this. So obviously, going to recommend using condoms. Although HPV, it's not perfect because the condom doesn't cover every area of the skin and it can still be transmitted. It's kind of too late here. Hopefully, this will clear itself up. But the practitioner recommends that Sean recommend to other people the HPV vaccine, particularly as a younger brother and sister, before they become sexually active. So this is an important point. Human papillomavirus has a DNA genome, as we said. There are many different strains. HPV infects about 80% of adults in the United States. So not all strains are linked with cancer. The biggest cancer risk generally is cervical cancer in women. Most strains are highly infectious. Any sort of sexual contact, including oral sex, can lead to contraction of this. So there are cases where oral sex can lead to throat cancers from HPV. Obviously, condoms is partial and that prevents other sexually transmitted infections. But there is a vaccine that can help protect, but it is only useful or effective before exposure. So kids entering their teens, this is when they should get this vaccine, and it will protect them against many strains of HPV, and it's particularly important to help reduce the risk of this virus transmitted cancer risk. So particularly that cervical cancer one. So hopefully higher vaccination rates will lead to lower cases of this occurring. The papillomaviruses are restricted primarily to the skin or mucous membranes only, so they cannot infect other tissues, so they won't move into the other systems of the body, which can make them less of a worry. But this cancer risk, right, the cancers that develop can metastasize and move to other areas of the body. Papillomaviruses have a replication cycle where they will enter by binding to a host cell receptor. So this is an epithelial cell here. On the surface of epithelial cells are all kinds of receptors. The double-stranded DNA virus has this outer protein, and it binds to the receptor. And once that happens, it induces endocytosis. So it gets brought into the cell. Then the virion kind of comes apart and releases the genome, which goes into the nucleus. The genome is uncoded in the nucleus, and it uses host DNA polymerases to replicate itself, right? So to copy its DNA genome. It also uses host RNA polymerase to make RNAs, which go out and get translated by the ribosomes. Those protein capsids come back into the nucleus, and they package up new virions. The virions are shedded through the epithelial cells as well. So what's going on with the cancer issue? Well, the virus can be eliminated sometimes. So this is why not everyone develops long-term infections or cancers. But in some cases, in the basal cells, it integrates into the genome. So it will actually integrate into the host genome, kind of like that lysogenic cycle. And when it's in there, it can either disrupt genes or it changes the expression of certain genes. We call these oncogenes E6 and E7. This is beyond us, but these genes are critical to cell replication. So when it turns them on, it turns on cell replication, which then leads to cancer. There are no drugs to treat papillomaviruses. We don't have any antivirals for this. So this one, very important to vaccinate children early in their life to prevent the spread of this. Hopefully we can decrease the rate of spread because of that. All right, so that's papillomaviruses. In the next one, we have influenza viruses, which have an RNA genome. We're going to talk about some ways that influenza can evolve rapidly. That's slightly different than what we've talked about previously. And then we'll, again, talk about the replication cycle. All right, so let's get some statistics on flu. Probably you have had the flu, I'm guessing. Normally, it is not a problem for the majority of people, but it infects about 10% of the U.S. population every year. There are about 36,000 deaths per year in the United States. This is mainly the elderly and the immune compromised who are dying from the flu. Just for comparison, the first 2 years of the COVID-19 pandemic, we had 352,000-ish deaths in 2020 and 460,000 deaths in 2021. I couldn't find accurate numbers for 2022, but I'm guessing it is lower because we have higher vaccination rates, people have been exposed, and unfortunately, most of the susceptible people have died. That is not to say that people are still not dying of it. But COVID will probably end up looking like the flu, where it's kind of always with us, and unfortunately will probably continue to kill the elderly and the immune compromised. Flu has a property called antigenic shift, which is like antigenic drift but can lead to larger changes. This rapid evolution leads us to have many, many strains of the flu out there, and that leads us to need new vaccines every year. There is a whole science to how the vaccines are made. They work to predict what strains of flu will be prevalent based on what they see in the southern hemisphere, which has a flu season that is opposite ours because the seasons are opposite ours. This whole thing, this antigenic shift, is due to the influenza virus genome being segmented. There are little pieces in there that can rearrange and recombine. They're sort of like short chromosomes. Not quite. I'll show you an image here. So the genome is segmented. So here's a flu virus, and each of these little squiggles is an RNA molecule. They're segments of RNA. Now, that in and of itself is not what leads to this rapid antigenic shift. What usually happens is there are different strains of flu that are primarily found in different organisms. We have avian flu. There's an outbreak of bird flu going on right now. I'm not sure what its status is, but we were very worried about it. It did make it to Idaho and several other states. There are strains of human flu, and there are pig flu strains. And these can kind of move between each other. Bad thing is that multiple flu strains can infect a single organism. So you might have a farm and you have birds like ducks and you have pigs and you work in your farm, and you could potentially transmit your flu virus, and a bird can transmit its flu virus to a pig. So the pig is infected with two different flu viruses at the same time. These RNA segments could be swapped between them and creating a new strain of virus. This is rapid evolution, and this is why we get such rapid evolution of new flu strains, this antigenic shift by what we call reassortment, basically. The strains are named for the spike protein, the HA spike, so H1N1, and the N is their neuraminidase, which helps it bud out of the cell. So the flu virus tends to infect upper respiratory cells. That's why we tend to get a cough and things like that with it, and it can cause respiratory disease. To enter the cell, the HA proteins on the envelope will bind to host cell receptors. That leads it to being brought in by endocytosis. The flu is a negative stranded RNA. It goes in and it can get converted to positive strand, which then helps make negative strands, which will be packaged up for transmission. The thing about RNA viruses is they tend to be very error prone. There are less proofreading steps in RNA replication. Influenza is very notorious for being error prone. It does not proofread its RNA replication, so that leads to new strains as well. The other thing that happens is that the RNA gets read and makes mRNAs, which then go out and get translated, making the various viral pieces, like these HA and NA proteins on the surface. The NA helps it get out. So everything gets packaged up and it moves to the membrane, where it's going to take a little bit of membrane with it, and it's an enveloped virus in this case. So we do have some drugs to combat the flu, and really these drugs should be reserved for people who are at great risk of dying from the flu, because obviously if we apply these drugs, we lead to selective pressures and we could have resistance evolve. So Tamiflu is one you've probably heard of. This helps block the viral exit, working on NA and things like that. So the neuraminidase helps cleave the host receptor that lets it out, lets the virus bud out. Okay, let's take a look at an animation of this life cycle.
The flu is a contagious respiratory infection caused by influenza viruses. An influenza virus attaches to a cell when hemagglutinin, one type of protein in its viral envelope, binds to a host receptor, a glycoprotein. The cell then takes up the virus by endocytosis. The endocytic vesicle fuses with a lysosome. The low pH in the lysosome causes hydrogen ions to leak through the virion's ion channel. The hydrogen ions cause a virion to disassemble and fuse with the endocytic membrane. The virion's contents are released into the cytoplasm. The 8 RNA segments of the influenza genome are covered with nucleocapsid proteins and are attached to several other proteins that make up the viral RNA polymerase. These RNA protein units enter the nucleus via the nuclear pores. The RNA segments are minus-strand RNAs, meaning that they are non-coding strands. Each of the 8 segments needs to be copied into a complementary plus strand by the viral RNA polymerase before it can be used for translating proteins. The new plus strands carry caps taken from host mRNA molecules. The new viral mRNA leaves the nucleus for the cytoplasm, where the host cell's ribosomes translate the mRNA. In this example, the mRNA codes for the nucleocapsid protein that will code future viral RNA molecules. Several other mRNAs code for viral envelope proteins. Envelope proteins are made by ribosomes on the host cell's endoplasmic reticulum. This particular mRNA codes for hemagglutinin. Back in the nucleus, the newly made nucleocapsid proteins prime and stabilize viral RNAs as they are being synthesized into full-length plus strands, this time lacking a cap and a poly-A tail. Newly made RNA polymerase proteins enter the nucleus and participate in the production of additional full-length minus strands that will serve as the RNA genome segments of new viruses. From these events, 10 different types of proteins are synthesized from 8 RNA segments, and new minus-strand RNA genome segments are produced. The RNA genome segments, complexed with RNA polymerase, nucleocapsid proteins, matrix protein, and packaging proteins, are exported from the nucleus. The envelope proteins made on the endoplasmic reticulum move by transport vesicles from the endoplasmic reticulum to the Golgi apparatus and finally to the plasma membrane. The RNA protein complexes are then packaged into progeny viruses as they bud from the cell membrane. The infectious cycle of influenza causes rapid destruction of mucosal cells.
All right, that animation, a little more in detail than we need, but I think it gives you a good understanding of how viruses get in. They have to do certain things to both replicate themselves and hijack the host machinery to do that replication. Then they need to package up new virions and escape from the cell. In normal cases, the flu infection is rapidly responded to by the host immune system. We haven't talked about the immune system yet, but your immune system, once it starts recognizing those viral spike proteins and things like that, starts making responses to it. Usually you're a little bit primed because you've seen other flu viruses, so it slightly recognizes them. But the general cycle is the virus replicates quite fast, but your immune system starts kicking in, and viral levels will drop down as your immune system is killing those infected cells and preventing viruses from replicating. This is not a long-term infection cycle like HPV was, right? You normally get back to normal in 1 to 2 weeks. So a very different kind of infection cycle here. Okay, so let's do a little review before we go on to the final section. Hypothesize which characteristic of the influenza virus makes the genome particularly susceptible to antigenic shift. Is it A, the genome is composed of RNA, not DNA? B, the capsid is coated with host-derived envelope? C, the genome is segment? Or D, the genome is minus sense? Go ahead and pause the video. Think about this one. Okay, the answer is going to be C. The genome is segmented. Remember, antigenic shift was when the segments were able to kind of recombine and reassort. So that segmented genome allows it to evolve so rapidly. These other things do allow it to evolve somewhat rapidly, but the segments are the biggest part. All right, our final virus, human immunodeficiency virus. You've probably heard of this one, also known as HIV. This is a retrovirus, which involves the process of reverse transcription. We'll talk about the HIV virion a bit, and then its replicative cycle and how that leads to a disease called AIDS. So here's a case study. We have a 24-year-old. He's been promoted at his car dealership that he works at, and he's ready to propose to his girlfriend, and it's been many years since he's had other partners or engaged in any risky behaviors, but he and his girlfriend both decide to get tested for STDs. This is where Ralph learns that he is HIV positive. It's surprising because he has no symptoms of this, and it's been years since he's engaged with any other partners, so this surprises him. We will learn what HIV does to the immune system, but the short answer is it infects T cells, which are critical to our immune system and fighting infections. So one of the things that we do when we discover that someone is HIV positive is we look at their T cell count. His cell counts are quite low, 500 cells per microliter, and normal levels are about 1,200 cells per microliter. So this is bad news because he is now more susceptible to infection. So he has a problem, how to avoid infecting his future wife and how to conceive healthy children. This is something they want to do. The first thing is controlling the viral replication. The current recommendation is that as soon as someone finds out they're positive, to start antiretroviral therapy. In this case, it's going to start a dual therapy, tenofovir, emtricitabine. These are both reverse transcriptase inhibitors. They're put together in one pill named Truvada, and so this one pill has 2 drugs, and he has to take this every single day. This is going to inhibit the process of reverse transcription, which this virus needs to replicate. This should hopefully halt that decline in T cell levels, and it will decrease the transmission risk. I am not an expert on HIV. The standard saying is that if the virus is undetectable by PCR, which you can get to by taking antiretrovirals, then it is not able to be transmitted. That is the standard knowledge, as far as I know. So they're going to work to bring the viral levels down. There is some controversy in the field of when to start these drugs, because HIV, being a retrovirus, is going to mutate quite rapidly. And once you start taking drugs, they are a selective pressure. They are going to cause it to evolve resistance. So to delay the side effects and to postpone resistant strains, some people say wait till you're below 500 T cells per microliter. So there's still differing opinions on how to treat HIV. The HIV virus itself, it's called a lentivirus. It's a retrovirus, which means it has an RNA genome that goes into the cell and gets converted into DNA. We'll see that process in a moment. It has a capsid around it, and then it has an envelope on it with spike proteins that help it trick its way into T cells, which is where it replicates. In the genome, there's this encoded protein called reverse transcriptase. This does the reverse of transcription. It takes RNA and converts it into DNA. This enzyme has a very high error rate. It does not proofread and makes lots of mistakes. That leads to rapid evolution here. This is the retro in the name retrovirus. It's not like they're throwbacks or anything like that. It's because they're converting backwards. The cycle for HIV, it infects T lymphocytes, so T cells, and it binds to this CD4 receptor and the co-receptor CCR5 on the surface of these cells. There are a few instances where there are people who are mutants that do not make these, and they are immune to HIV because of that. That's another deeper story. Once it binds to these, it gets incorporated into the cell. Once it gets into the cell, reverse transcription happens, so the RNA gets made into a double-stranded DNA. Now, here's the I guess, deadly part about HIV. HIV's DNA can incorporate into the host genome. So once you have been infected, its sequence is in your genome forever. You cannot be rid of that sequence. That is why treating HIV is a lifelong prospect, because the viral DNA is encoded in those cells and it's there forever. That DNA gets converted into RNAs through transcription. They get processed into new virions, and it can leave through budding. And in this process, it infects other T cells. So the thing here is not the virus killing the T cell. The T cells actually get killed by the human immune system. They recognize that they are infected with a virus, and our body actually kills those cells. But those cells are needed for proper immune system function. So as their level drops, this leads to a weakened immune system, which can lead to acquired immune deficiency syndrome, or AIDS. AIDS is the disease caused by HIV. AIDS opens patients up to secondary and opportunistic infections. So we talked about toxoplasma, the eukaryotic parasite. That can then infect these people, and their immune system is not able to respond to it. So it grows and grows out of control. The flu can be very deadly for them. All kinds of infections that normally are not deadly to a person with a healthy immune system can become deadly to an AIDS patient. Okay, we're going to watch the animation on this one. It'll explain it much better than I can.
The human immunodeficiency virus, HIV, is the causative agent of acquired immunodeficiency syndrome, or AIDS. HIV has an RNA genome surrounded by a protein capsid, which in turn is surrounded by a membrane taken from the host during virus exit. Embedded in the membrane are virus spike proteins needed to attach and enter host cells. The virus capsid contains the enzyme reverse transcriptase, which will copy the RNA genome into DNA during replication. The first cells infected by HIV are T lymphocytes, or T cells, that possess cell surface proteins called CD4, which is the receptor, and CCR5, which is the co-receptor. As an HIV virion binds the CD4 and CCR5 receptors, its envelope fuses with the cell membrane of the T cell. The HIV core is released directly into the cytoplasm. HIV is a retrovirus, a type of virus that uses reverse transcriptase to copy its RNA genome into DNA. Double-stranded DNA is produced, a process that involves cleaving away the original RNA template and replacing it with DNA. The double-stranded DNA enters the nucleus through a nuclear pore. A viral enzyme integrates the HIV DNA at a random position in a host chromosome. The integrated DNA is called a provirus. Integration is a critical step in that it ensures permanent infection of the host. The entire length of the integrated HIV genome is transcribed to RNA by the host RNA polymerase. Some of the RNAs become the RNA genomes of new virions. Other RNA molecules are spliced and translated to form viral proteins such as GAG capsid proteins and ENV spike proteins. The spike proteins are made on ribosomes that dock on the endoplasmic reticulum. Transport vesicles bring the proteins to the Golgi apparatus for packaging before they are exported to the cell membrane. At this stage, the protease, which is part of the long GAG-Pol protein, cuts itself out of the polyprotein. The protease also cleaves the polyproteins at other sites to release the core proteins and enzymes. The protease activity is required to generate infective virions with a conical core structure. Protease is the target of important drugs called protease inhibitors. An infected cell can release a large number of new virions. An alternative to viral budding is cell fusion, mediated by the binding of the spike proteins in the membrane to CD4 receptors on a neighboring cell. The fusion of many cells can form a giant multinucleate cell called a syncytium. Cell fusion enables HIV to infect neighboring cells without exposure to the immune system.
All right, so again, more detail than we need, but I think it's really important to see what is occurring in there and how, I guess, simple the process is, but that makes it difficult to treat, right? There's very few steps in there, and that integration into the genome, that's game over at that point. So we'll talk about this later when we get to the immune system chapter, but I wanted to introduce it here. The way an HIV infection occurs is generally we have an amount of T lymphocytes, right? Those CD4 T cells here. Normally you're somewhere up in this range, and then in orange here we have levels of the virus. So initial infection has high replication of the virus, and there's usually a dip in T cells. So there is usually some sort of fever and illness associated with the early stages in the first few weeks of HIV infection. But then viral levels drop, and it lays kind of dormant for several years. And it's not until several years later that we start to see viral levels increase, and we don't start to see any sort of disease symptoms and things like that. So we start to see weight loss, fatigue, other opportunistic infections occurring. We don't start to see those until much, much later. So if we think back to our case study, that individual was shocked to see that they were infected because they didn't have any of the symptoms here. But as the disease progresses, the T cells continue to go down. Because of this, because of that viral integration, we have had to come up with antiviral drugs. And probably HIV is one virus that has probably the most drugs out there. The antiretroviral drugs are going to target important points in the replication cycles to prevent the virus from replicating. HIV is at epidemic levels in the U.S. Since the 1980s, we've had over 700,000 people die, and of course, many, many more are infected. Again, though, I want to put those numbers into perspective. When we compare that to the COVID-19 pandemic, more people have died of COVID-19 than have died of AIDS. So this is something that is livable with now that we have drugs. We have several drugs here. We'll talk about AZT. That was our first drug for it. It's a nucleoside. It's a base analog, basically. It halts elongation. We have several of those. Fusion inhibitors, preventing the viral fusion from occurring. Blocking the spike protein from binding to co-receptor CCR5. Binding to reverse transcriptase and inhibiting that. Most of our cells don't undergo reverse transcription, so we can block that. Blocking integration of the DNA genome, protease inhibitors, as it was mentioned, stopping that cleavage from creating the mature virion. This is going to have side effects because our cells use proteases for lots of things. Same thing with the base analogs. These are going to have side effects because we need those bases for our own DNA replication, so growing cells might have some issues. So let's talk about AZT, our first drug to treat HIV. This is just a small number of the drugs that are out there as well. There are many, many more, and we use them in combination oftentimes. So AZT was our first anti-HIV medication. It is a modified nucleotide. Instead of having the 3' OH, it has an N3 group there, which, when it gets added to a growing chain, so this is growing 5' to 3', when it gets added, no new nucleotides can be added to this base. So it stops the growing chain, prevents DNA replication from occurring, and that prevents viral replication from occurring. Now, this is going to have side effects because it's also going to prevent human nucleotide replication from occurring. So it is not the best choice of medication, but still can be used. It is interesting to note that HIV is just one of many retroviruses that are out there. Many of them don't cause this dramatic disease as this one. But when we look at the human genome, we notice that a large portion of our genome actually looks like retroviruses that have integrated with our genome. And over the years, they have lost the ability to move out of the genome. So we call these retroelements. Up to half the sequence in the human genome might have viral and retroviral origins. So it's really interesting to think that you have a large amount of viral DNA in your genome. And there are implications evolutionarily for that are beyond this course, but just something interesting to think about, that you are part virus because of this process. So here's some chromosomes, and all these little blue and red and green bits are things that we think are viral sequences in there. Okay, that covers Chapter 12. Viruses are fascinating. I love to talk about this topic, but we only have so much time in this course. But I hope that gives you at least a broad understanding of what's going on. I want to reiterate that viruses are very different from the other organisms we talked about, and they are not affected by our antibiotics. Antibiotics work on bacteria. We need other drugs for viruses. Those are called antivirals. All right, that's it for Chapter 12. Bye for now.